Okay, so part of my charge is to keep things moving on time, so it's on time, so we're going to start on time. This is the first of two related presentations about some research that was different aspects of it were done by different students. And so Dr. Gold is going to say a few words about both of the students' research after the second presentation. They're one right after the other, and bestow the honor stole on both of them. Matthew will give his presentation, and we'll have some time for questions and answers, and then we'll be Nicholas' turn. So this is Matthew Fensl, mentored by Dr. Daniel Gold, and his talk is titled Determining Transmission of the Parasite Labaricola migrator in the laboratory colony of Rhamphadorina portentosa. I hope I can't quote.
I'm sorry.
Okay.
Hey everyone, my name is Matthew, and today I'm going to be talking about some of the work that I've been doing to help determine and sort of enhance the transmission of this Gregorian parasite Labaricola migrator in the Madagascar missing cockroach. So first, just to sort of set the stage a little bit, I want to talk about parasites and some of the burden they can have on hosts. So parasites are organisms that live on or in a host organism, and they will take up resources that can be better used by the host, and so this is what causes a disease in the host or other symptoms. So protozoan parasites, which is kind of important for this talk, are microscopic single-cell organisms, so they're a particular type of parasite. And there are a few examples of this that you may have heard of. So the Plasmodium parasite, which is a genus of parasite that causes malaria. You also have Giardia, which causes the disease giardiosis, which is a diarrheal disease. Both of these diseases have a lot of impacts on human health, so they're really studying parasites. Again, the Plasmodium parasite will cause malaria, and they do this by infecting the blood cells of human hosts. So if you see this image here, these are red blood cells from a human host. If you look here, it's just a normal red blood cell, and what you're seeing here is a red blood cell that's been infected by a Plasmodium, one of the parasites in the Plasmodium genus. So it's sort of a particular ring structure, and it will carry out its infection and reproduce in the red blood cell of the human host. And what you're seeing here is sort of the proliferation of this parasite in the red blood cell. And eventually what it will do is actually lyse the red blood cell of the host organism. And so all these replicated parasites can now go out and infect other red blood cells. And that's sort of where you get the pathogenicity of malaria disease, because it's lysing the red blood cells of the host. Now, to speak more about Gregorians, they're this really understudied group of parasites, which is really interesting because they're actually quite diverse, and hopefully I can kind of drive that point home. So they're a subclass of Apicomplexan alveolar parasites like invertebrates. So branching out from protozoa, you'll see these are Apicomplexan, and you'll see Gregorians over here in this blue area with there's sort of three distinct groups of the Gregorian parasite. Now they're extremely abundant and diverse. About 250 genera and over 1,600 species of Gregorians have been identified so far, and they actually have a lot of really novel sort of structures within the organism rather, that are really interesting, haven't been seen in other parasites. However, they're actually really understudied, and not much is really known about sort of the phylogenetic relationships between different Gregorian parasites. To sort of speak to this, only two species of Gregorians have a genome sequence, that being Gregorina nefandrines and Astrogregorina thailensis. And a lot of this is mainly due to the fact that since they are infecting invertebrates and not vertebrates such as humans, there's not really as much of a medical interest in studying them. And so the little research that has been done into Gregorians is mostly focusing on the phytology, sort of like the evolutionary relationships between organisms, and not really what we wanted to get at, which is sort of the cellular and molecular mechanisms of how it's affecting the host organisms. And something I just want to highlight here really quick. So I mentioned before that this is where Gregorians branch off from complex up. And you'll see above that Cryptosporidia, which is home to the Cryptosporidium genus of parasites that cause cryptosporidiosis, another diarrheal disease. In the next talk, my colleague Nick Barrett will be talking more about Cryptosporia and sort of how they relate to the work that I'm doing here. So again, talking about Gregorian host diversity, they've been found to infect a wide range of organisms. So that includes insects, crustaceans, arthropods, mollusks, bivalves, and gastropods, just to name a few of the different groups of organisms that have been found to infect. And why this is really important is because just to even consider with insects, some scientists believe that some of the most diverse groups of organisms on the planet are parasitic wasps and beetles. And because Gregorians could potentially infect all of these different organisms, they may very well be the most diverse organisms on the planet, or at least one of the most diverse groups of organisms on the planet. So the fact that they're so understudied is really interesting considering this. To sort of speak to that even, less than 1% of all living invertebrates have actually been examined for the presence of Gregorians. So again, there could be many more organisms that are affected by Gregorians, we just don't know it. Now, looking at Gregorian host interaction, Gregorians will inhabit different body spaces within the vertebrate organisms that they parasitize, mostly the intestines of the organisms, but sometimes coeloms and reproductive vesicles, just other structures within the body, but again, mostly the intestines. And so what you're seeing here is a sort of simplified diagram of the life cycle of Gregorians within an invertebrate organism. And so I want to highlight just a few stages here that are really important to the research that we're doing, that I'm doing specifically. So we have here the gametocyst. Now this gametocyst exits from the frass or feces of, in this case, the Madagascar hissing cockroach, and contained within this gametocyst, now that it's in the environment, are what are called these oocysts. And so what happens is the oocysts will sort of escape out of the gametocyst into the environment, and within the oocysts are sporozoites. Now, just make a quick differentiation. The oocyst is the infective stage of the parasite, so that's what's actually being taken up by a host organism such as the hissing cockroach. And the sporozoite is actually the invasive stage. So what you're seeing here is once the oocyst gets to the bowels, the intestines, there'll be cellular signaling that allows for the sporozoite to actually exit the oocyst. And what it's doing is partially invading the intestinal epithelial cells of this host organism. And from there, it's able to carry out its life cycle, which I won't go into here, but again, I just want to highlight these few life cycle stages right there. Now, most known Gregorians are host and stage specific, and what I mean by that is the host organism, so like the particular organism that's infecting, but also the different stages. And in the case with like most insects, they'll have different stages of their life cycle, and so you can actually, interestingly enough, have different species of Gregorians infecting those different stages. And so, to speak to that, when we first started our research into Gregorians, we were trying to use the mealworm beetle, or Xeniber molitor, as a sort of system for helping to analyze, to do research into Gregorians, And why we kind of focus on this one first is because research has already been done using these organisms into Gregorians. And actually, something interesting is that 4 different species of Gregorians will infect different life cycle stages of this organism. So in the juvenile stages, there'll be 3 different ones that will infect it, but in the adult stage, only 1 particular Gregorian will infect it. So that was something interesting. We wanted to get at why is this occurring, what mechanisms are at play that might affect the different species that are infecting different stages. And so now what you're seeing here is just a life cycle of the migrator in the Madagascar hissing cockroach. And again, I just want to highlight the oocysts are the infective stage of the parasite. So what's happening here is when the gametocyst dehisces, it's ejecting what are called these spore tubes that you're seeing here. And what that's doing is increasing the surface area that the parasite is covering. And so from there, the oocyst can be taken up by the host organism and again contained within the gametocyst. it will get to the intestinal epithelial cells. Now, the gametocyst is interesting because it's actually incredibly environmentally resistant, as are the oocysts. So in this way, it's sort of serving to really protect the actual invasive stage, the sporozoites of the parasite, so that they're not subject to environmental stresses. Now, what actually causes them to dehisce there's several factors such as humidity, temperature, and that's what's causing the dehiscence of the gametocyst. And again, sporozoites will create the intestinal epithelium. Not sure why that was. Anyways, the gametocyst of the migrator. So the gametocyst will dehisce when mature, releasing oocysts into the environment, and again, the oocysts are consumed by the host organism. So what you're seeing here are several gametocysts that we were able to isolate from the frass of one cockroach. And you'll notice here a scale bar with the gametocyst labeled there. The gametocysts will range in size from about 700 microliters to around 1,000 microliters. So this one's just shy of about 1,000, so it's about an average size gametocyst. So that's why that looks like that. And again, just to remind you, this is the gametocyst, what you're seeing here, and this is going to be the stage that is outside of the host organism as opposed to these stages which are inside. So again, as I mentioned before, we wanted to get at more of the cellular and molecular mechanisms of infection of these parasites. And so to answer these kind of questions, we needed to have a consistently infected population of host organisms that we could utilize. So one of the issues that we kind of ran into with our research was that we weren't able to definitively say that we could isolate gametocysts every time we were trying to carry out the process, which I'll get into here shortly. And so that's just a large time sink because it's just a very laborious process. It takes a lot, not laborious per se, but it is taking a lot of time to do this. And so it was just putting a stop to a lot of our research. And something else that we also, other research is going on in our lab is we also want to determine if bee migrator specifically is susceptible to this antimicrobial agent paramomycin. Paramomycin is used to help treat cryptosporidiosis, which is again the cryptosporidium parasite. And so we're trying to see if we can have an effect treating it with that particular agent. Now, using the hissing cockroach as our host organism, there's a few pros and cons, which I'll get into here really quickly. So some of the pros are that they're very easy to culture and they have a pretty long lifespan, about 2 to 5 years. And they produce these large grass or feces pellets, which are really good for us because they're easy to identify and you can manipulate them a lot more easily than, say, the frass pellets of the mealworm beetle, which was, again, was one of the issues why we started using the hissing cockroach instead. And also, only one recurrent species is infecting all of the life stages. As I mentioned before, with the mealworm beetle, there's different species infecting different stages. That's not the case here. There's only one infecting each stage. Now, some of the cons that are associated is that it has a long life cycle. It takes about 7 to 9 months for it to mature fully. So initially, we were looking more at the adult stages, and we were focusing on that. So this long life cycle was kind of an hindrance. And also, this particular system was better established for ecological research. So there wasn't really a lot of previous research that had been done, kind of looking at some of the questions that we wanted to try to answer. So it was just time consuming to find out some of the procedures that we needed to use to do this research. Now, the question that I specifically wanted to ask is it possible to effectively transmit bee migrator? So a critical step into research of bee migrator is enhancing the infectivity of the hissing cockroach. Again, I kind of identified that was a problem that we were facing. We couldn't, with any certainty, say that we were going to isolate the gametocysts each time we were trying to carry out that procedure. So some of the questions that I wanted to ask were, if food was laced with oocysts, which is the infective stage of the parasite, when cockroaches then become infected, well, the nymph stage of the hissing cockroach would be easier to infect, maybe because of developmental processes that are ongoing, it would be more susceptible. And could previously infected cockroaches that are not currently showing signs of parasitism then be reinfected? So these were just some of the questions I wanted to look at. So from Dr. Richard Clopton, we were able to utilize what he calls a frass plate technique. And what he does and what we do is that you will put individual cockroaches into individual glass plates, as you see here, and we allow them to sit for about 24 hours, and while they're doing that, they're hopefully defecating, and we're able to isolate those frass pellets, which is what you're seeing here. Now, in this image, they're being suspended in this Ringer solution that is meant to sort of simulate the pH conditions that you'll find within the inverter post. And so what we're doing from this point is we're actually teasing apart the frass pellets under a microscope to try to see if we can isolate any gameosis from the frass. And in this instance, we're able to do that. You'll see here's gameosis actually right there that we're able to isolate from the frass of the cockroach. Now, I developed, I sort of came up with this procedure that I could hope to sort of enhance the infectivity because, again, as I mentioned, that was an issue. So first, I was allowing the gameosis to dehiss, where they're, and you're seeing here the spore tubes being ejected out containing the oocyst. And what I was doing was allowing them to dehiss in this solution, the Ringer solution that I mentioned previously. And from there, I was extracting oocyst and weighing out equal amounts of food, and doing that because I was putting individual amounts of food laced with the oocyst into these individual dishes that we were using for the frass plate technique. So I had two groups: ones that I was infecting, and ones that I was using as a control, where I was not lacing their food. And so as you're seeing here, I'm lacing the food with hopefully the oocyst that come about from dehissing, from the gameosis dehissing in the solution. Here there's no manipulation, these are the controls. And after about 48 hours, hopefully they've ingested some of the oocyst that I've laced their food with, and you'll see frass pellets right there. So then we analyze that using the technique that I was talking about before. So I've only conducted a few rounds of this procedure so far, and so this is just some of the preliminary data. So from the first round of this experiment, what you're seeing here is on this bar is the gameosis that I was yielding in the different cockroach populations. So in the first round, I actually only was able to isolate the gameosis from my control organisms. These were the ones that I was not lacing their food, whereas the experimental ones where I was lacing their food, I wasn't able to get any gameosis. So, okay, that's the first round. In the second round, however, I was not able to isolate any gameosis from my populations. So while this is only 2 rounds of this procedure, perhaps it shows that maybe it's not the most effective for actually enhancing the infectivity of our organisms. But there is a few issues with my procedure. Perhaps maybe I'm just not allowing a long enough period of time for them to actually ingest the oocyst and then carry out the life cycle stage. Actually, I'm sure that's probably what it is. But there's a few other factors that I can tweak as well. So some of the future steps that we want to take are to conclude that if this direct infection procedure will enhance the infectivity of our organisms, and then compare the gameosis yield between colony types. So we'll have ones that are infected and ones that are uninfected, and hopefully we see, perhaps we'll see a difference between those 2 types. Determine if the nymphs are more susceptible. This is actually a big question that needs to be answered. Will it, because of, again, maybe some of the development processes that are going on, they might be more susceptible. We don't know. We need to figure that out. And finally, to see if some of the previously infected cockroaches can then be reinfected again. So I'd like to acknowledge Dr. Gold and the Gold Lab, the Hook Fellowship, and Dr. Richard Kaufman of Peru State College. And if you have any questions, I'd be happy to answer.
Got a couple of minutes for questions.
So why should I care if you can treat these guys with that antimicrobial and cure them?
Well, because they have been shown to be effective in treating cryptosporidiosis, caused by the cryptosporidium, seeing if maybe we can use this drug to treat them, we could then potentially have colonies that are totally uninfected by the parasite. But we need to see if this this drug has any adverse effects on the cockroach itself. But in doing so, we could then have sort of clean populations that aren't infected by the parasite.
So it's mostly to help you guys in your research to have.
Yes, ones that we can say definitively are.
Is there any thought that this might be a model for how to treat dysphoria in the future?
Oh yes, definitely. My colleague Nick can get into that more, but yeah, definitely. They could potentially have that role, so it's something that we want to look at.
Let's see this one with these guys.
Yes, certainly.
So genetic reason to use this drug?
Well, also maybe to see the effects that it has on sort of transcription of genes in the host organism.
All right, well, thank you.